Edgar Vega is the Senior Manager of Patient Advocacy for uniQure. uniQure is looking at AMT-130, an investigative gene therapy for Huntington's disease that is intended to silence the Huntington gene with the goal of inhibiting the production of the mutant protein. Edgar has worked in the healthcare space for the past 16 years, and he loves educating the communities he works with. So, it's you.
No, definitely. Thank you so much for the introduction. I had mentioned previously, you're all probably used to a little box with my face on it. So yeah, so I'm Edgar. And then I actually have a pre-recorded video with me that I sent to Michael. So hopefully we'll pull that up, and then I'll be around for questions afterwards, updates as well. So
thank you, Edgar.
hi, everyone. My name is Edgar Vega, and I am the Senior Manager of Patient Advocacy here at uniQure. And today we're going to be talking about our HD gene TRIX1 trial. And we're super excited. But before we do that, we're actually going to introduce a little bit about patient advocacy and what it is that we do here at uniQure. So come along with me as we do this. First of all, there's a little bit of a disclaimer. So just as we know, we're going to be talking about a clinical trial. So it's important to highlight that this information is just being presented in response to provide education to a lot of patient advocacy organizations, and then also that the safety and efficacy of any of AMT-130 that you hear in this presentation has not been established, and FDA approval has not been granted as well. So like I had mentioned before, my name is Edgar Vega, and I'm here, and I'm the Senior Manager here at uniQure. So on behalf of the HD disease team here at uniQure, I'm honored to really introduce our company and our patient advocacy department to all of you. uniQure has a long history in developing gene therapies, but it's a relative newcomer to the HD community. And so I'd like to provide a little bit of a background, to begin. So uniQure was founded originally as Amsterdam Molecular Therapeutics in the Netherlands in 1998. And in 2012, that innovative work of research there resulted in the first gene therapy ever approved in Europe. And this experience laid the foundation for our leadership today in the discovery, clinical development, and manufacturing of gene therapies for rare diseases. And so we currently operate out of 2 locations with all the science and research and technology that's in Amsterdam. And so you'll see that on the left-hand side. And then all the manufacturing and development being done just outside of a city in Boston called Lexington, Massachusetts. And a lot of the other functions, so clinical and regulatory affairs, are also spread out amongst both sites in Amsterdam and Lexington, Massachusetts. And one of the offices that's out of Lexington is patient advocacy. And here at uniQure, we really do try to with our focus on gene therapy research, we really want to ensure that the voice of the HD community runs through the DNA of our clinical programs. And so we want to understand the impact of the diseases that we're making products for and attempt to engage the communities that we work with, right? So we want to make sure that as we understand the HD community, we can take that information and help shape internal programs and hopefully create opportunities for that patient voice to be heard. And so patient advocacy really is an integral part of our organization. And so we're super excited. We want to have that impact on that scientist that's working on a vector in Amsterdam, and hopefully they can create that connection of working on something that's truly important. And so with that, I just want to introduce you to our 2-person team here at uniQure that is doing patient advocacy. And Daniel Leonard, you may be familiar with him. He's our Senior Director of Global Patient Advocacy. So he has responsibilities worldwide dealing with the Huntington's disease organizations. And then also me, Edgar Vega, and I have responsibilities here in the US and in Canada as well. Our goal really is to deliver one-time treatments that transform patients' lives, and those disease modifying approaches for several indications, Huntington's disease being one of them. And so with that, next I'll introduce Dr. Joseph Liberman, where he'll explain what gene therapy is and how it could work specifically in HD. And he'll also talk about our clinical trial in Huntington's disease as well, a little bit more in depth. And so I wanted to thank you all for your time and introduce you to Joe. Thank you.
Hello. My name is Joe Liberman, and I'm an Associate Director of Medical Affairs here at uniQure.
I'm really excited to be able to share our work on the HD GeneTrix-1 clinical trial. For those diseases where there is a genetic link, gene therapy represents a very promising therapeutic approach. Gene therapy is defined as a technique that uses genetic material to treat or prevent disease. First, we must figure out how to get the treatment to where it needs to go. Now, viruses already do exactly that. They find specific cells and inject their genetic material. So what our scientists have done is to modify a viral capsid so that it delivers our therapeutic genetic material instead of the usual viral DNA. The viral capsid can be compared to a box that might be delivered to your door. Then the scientists have packaged the therapeutic genetic material into an expression cassette, which is like an instruction manual or recipe book. Once the patient receives the package, then the patient has the instructions so that the patient's own body can continually produce the therapeutic molecule encoded in the DNA expression cassette. In the case of brain diseases, if the gene therapy is delivered to the brain, then the brain itself can be the factory that produces the therapy to correct the genetic defect. As this audience knows, the cause of HD is a mutation in the Huntington gene. With increased CAG repeats, the mutant Huntington mRNA produces mutant protein. This leads to clumping of proteins and degeneration of neurons across the brain. This degeneration is particularly concentrated in certain brain areas, which in turn is associated with the symptoms of the disease. For HD, uniQure is developing an investigational gene therapy approach to reduce or knock down the amount of mutant Huntington protein. It's important to highlight that the safety and efficacy has not been established, and there is no FDA approval. uniQure's gene therapy approach aims to reduce the amount of HTT mRNA, which is intended to reduce the amount of toxic HTT protein, aggregation, and neurodegeneration. The gene therapy is based on the box, AAV5, the instructions, an artificial microRNA against HTT, or MIHT, and it has the company name AMT-130. It is important to note that AMT-130 is investigational and has not been proven to be safe or effective. It is not approved for market by any regulatory authority. Let's take a closer look at AMT-130. This investigational treatment is the first one-time administered gene therapy to enter clinical testing for the treatment of HD. AMT-130 is administered only once by a neurosurgical procedure. There are two key components to AMT-130, an AAV5 vector and a gene encoding a microRNA. The vector acts as a delivery system and is based on a non-disease-causing adeno-associated virus, known as AAV5, that has been changed to carry and deliver a gene-encoding microRNA that will recognize, bind, and non-selectively lower the human Huntington protein. MicroRNA, or miRNA, are small pieces of genetic material that can prevent production of a given protein. In this case, to non-selectively lower human Huntington protein means that production of both the disease-causing mutant mHTT and normal Huntington protein, HTT, could be decreased. We'd also like to add that AMT-130 is investigational and has not been proven to be safe or effective and is not approved for the market by any regulatory authority. The animation shows where AMT-130 is administered in the brain. In this video, a brain is shown with the left hemisphere removed to show the deep gray matter structures in tan and the CSF-filled lateral ventricles in light blue. The neurosurgeons will infuse AMT-130 directly into the caudate and the putamen bilaterally under the guidance of MRI. Then MIHTT spreads to other regions associated with the neuropathology of Huntington's disease. So we are calling this gene therapy study HD GeneTrix-1. The objective of this phase 1/2 clinical trial are to assess the safety, tolerability, and efficacy of AMT-130 in patients with HD using a dose-escalating, randomized, and double-blinded control design. The inclusion criteria include, but are not limited to, patients with a definitive clinical diagnosis of early manifest HD, genotype of 4 CAG repeats or greater in the Huntington gene, between the ages of 25 to 65 years old. There are several sites open and recruiting in the United States. Please note that participants will need to be off any other experimental agents for 60 days prior to enrollment. Previous exposure to gene therapy, Huntington lowering strategies, or experimental brain surgery are prohibited. It is important to reiterate that AMT-130 is administered only once in this study. The study is testing two dose levels of AMT-130, a lower dose and a higher dose. The safety of the low dose will be assessed before testing the high dose. A total of 26 patients will be enrolled in cohorts 1 and 2, 16 of whom will receive AMT-130, the treated group, and 10 will not receive any study treatment, the imitation group. Following completion of the first 26 participants, we intend to enroll up to 18 additional participants in cohort 3, where the goal will be to evaluate surgical techniques that simplify and shorten aspects of the administration. Patients assigned to the treated group will receive the dose of AMT-130 during a neurosurgical visit. AMT-130 will be infused into two specific brain regions, the caudate and the putamen, under general anesthesia. This is done by drilling 2 to 6 small holes in the skull and administering AMT-130 by a microcatheter. For patients assigned to the imitation group, similar scalp incisions will be made under general anesthesia, and the patient will remain in the operating suite as if the full administration procedure were being performed, but no holes will be drilled into the surface of the skull, and they will not receive a dose of AMT-130. The main part of the study lasts for 12 months, with additional visits out to 5 years for continued safety follow-up. Procedures will include clinic visits, assessments of physical and neurological health, a neurosurgical procedure, lumbar punctures, brain scans, and samples from body fluids. The study is double-blinded, meaning neither the patient nor the investigator or clinical staff will know if the patient is in the treated group or the imitation group. Participants who are randomized into the imitation group may be eligible for treatment with AMT-130 in a crossover group after completing the month 12 blinded follow-up visit, provided that one, the Independent Safety Monitoring Board permits a crossover study based upon the benefits and risks of treatments as noted at that time. And two, the patient still meets all study criteria, including volumetric MRIs. Since this is the first study of AMT-130 in humans, many questions remain unanswered, such as how long the effects will last or what the long-term effects of AMT-130 are. As I said previously, safety and tolerability of AMT-130 and its administration is being investigated in the HD GeneTrix-1 trial. The variability of response across patients is also something that is being investigated. Since AMT-130 is investigational and has not been FDA approved, there may be side effects or risks that are not known at this time. Please feel free to contact your nearest clinical trial site or your healthcare provider with questions about gene therapy in general or our HD GeneTrix-1 study. 2021 was an exciting year for AMT-130. In April 2021, uniQure announced completion of enrollment in the first cohort of the phase 1/2 clinical trial of AMT-130 for the treatment of Huntington's disease. Enrollment of the second dose cohort in HD GeneTrix-1 is also now complete. We are adding a third cohort to examine the possibility of administering AMT-130 in a slightly different way. uniQure has also announced plans to initiate a second clinical study of AMT-130 in Europe in the first half of 2022. This study will be called HD GeneTrix-2. We are so grateful to all our investigators, our collaborators, and especially all the patients and their family and friends. Everyone has poured their hearts and souls into this study so that we can all better understand the effect of AMT-130 on Huntington's disease. Here's the contact information for our sites in alphabetical order by state. The first 6 are at the University of Alabama at Birmingham, University of California, San Francisco, University of Florida, Rush University in Chicago, Johns Hopkins University in Baltimore, and Beth Israel Deaconess Medical Center in Boston. Our next 5 sites are at the University of Michigan, Ohio State University, Vanderbilt University, as well as the University of Texas at Houston, Virginia Commonwealth University, and the University of Washington Medical Center. If you or someone that you know might be interested in participating in our study in the United States, please do reach out to our clinical trial sites. And for the most up-to-date information on trial sites, please visit hdtrialfinder.org, clinicaltrials.gov, or write to us directly at AMT130_clinical_trials@uniQure.com. We look forward to hearing from you.
All right, perfect. That video was shot a couple months ago, actually probably about close to about a year now. And so since that time, there's been a couple of updates. We heard Joe mention about the European trial. So that actually did kick off in the second half of 2022. And we enrolled the first 2 cohorts pretty easily in Europe. Since that time, we're doing pretty well. There were meetings with the Data Safety Monitoring Board, which kind of monitors the safety of the trial. And then in August, they had met in June of 2022. In August of 2022, we were made aware of some, they call them SUSARs, which are serious, unexpected, severe adverse reactions. And we were made aware of 2 cases in the European trial of some second-hand effects that Joe had mentioned that could come up. And so then at that point in time, we made the decision along with the DSMB to pause enrollment in the trial. And this was just seen in the high dose cohort. If you remember from the video, there were 2 different groups, one group that received a low dose and then one group that received a high dose of the medication, because this is a dose finding study. So we're trying to figure out exactly what the appropriate dose is to have a therapeutic effect in the trial. And so the trial was paused. There was an investigation that was done during that time. And then the DSMB reconvened in about October and actually allowed the trial to start enrolling again with some changes to some criteria. Small changes, but important changes. So there was an MRI that was done 2 weeks post infusion. And so now that got changed to 7 days, right? So little small changes in order to just make sure that we're not missing the safety aspect of it. That's where the trial stands right now. The 3rd cohort here in the United States is actually subject to a change in the way that the actual surgery is being done. So to make it a little bit more efficient and quicker. So right now, the expected is anywhere between 8 and 12 hours. So they really want to try to cut that down for that 3rd cohort. And we expect that the 3rd cohort should hopefully start enrolling at some point during the first quarter of 2023. But we haven't actually heard that the trial sites are enrolling the 3rd cohort yet. And same thing for Europe as well. So that's where the trial stands for right now in terms of updates. I'm trying to think of what else I can give you an update on. And that's really pretty much it in terms of like updates. So I don't know if there are any questions or anything like that you had received previously or anything, but if not, thank you so much for your time.
Yeah, I have a question, Edgar, and I'm not sure if this is something that can even be discussed, but that safety trigger, was that like so someone had an adverse effect? Is that what that triggered that safety?
Yes, and actually, exactly. So it was post-infusion. So in Europe, it's being done a little bit different. There is no placebo group in Europe. And so patients are being dosed in a high dose cohort and then a low dose cohort, right? And so the participants know that they're going to get the drug. Here in the US, the FDA wanted a placebo group. And so that's why there's that change. And the participants know that they're in the group. And so there was some effects that, looking back at it, were more like procedure related, right? And not necessarily attributed to the drug. And so those 2 participants have had full recoveries since that pause in that trial. And again, just these safety changes have been made in order to make sure that we caught things a little bit earlier rather than 2 weeks, or which was the case in these 2 cases. And then after the fact, they went back and they looked at a lot of the information from the stuff that we had, and there had been an adverse reaction in the US high dose cohort trial as well. And they reclassified that as including that. So the total, there was 3 cases where we did see some of those adverse reactions. But again, it was just like a looking back and, oh, these symptoms sound very similar. Let's just include it in that safety profile of the trial.
And then with all 3 cases, the same adverse reaction?
Yeah, we saw headaches, trouble walking. I think one of the participants had some trouble like speaking for a little bit. And so they readmitted those patients over to the hospital, saw that there had been some improvement. And then as time went on, the participants improved overall.
And seeing as how there is that placebo group in this, which is, I think, crazy that Europe said, because it still costs the same amount of money for a sham surgery, right? Basically, less people are able to get it in the US to participate like in data than Europe with the actual on drug. But my question for that would be, so do the people have to wait? How long do they have to wait to be unblinded? Is this like they have to wait till the very end? Because I know that uniQure has done these like cohorts, right? It's not just like, oh, this is phase 1 and these are all the people. Like it's been very slowly rolled out for safety, I imagine. So when do patients get unblinded?
If you're in the placebo group, you get unblinded 1 year from enrollment, right?
Okay.
And then at that point in time, you are eligible at that point to be put into the high dose or the low dose cohort group, treated group. The one caveat with that is that you would have to re-qualify for that for that actual treated group to be randomized into either one of those groups. And again, that's the one caveat about being in the placebo group, right? What we are finding is a lot of patients will meet that criteria, but sometimes it'll be something as small as like brain volume, right? Because we need to make sure that there's enough actual brain matter for the drug to actually hopefully have an effect. And so sometimes we've had some trouble with some of those, that inclusion exclusion criteria, right? And again, it's just a matter of requalifying for that high dose or low dose cohort.
So if they are in the placebo group, they have the option to get the drug like an open label extension?
After 1 year, yeah. So
After 1 year.
yeah, and then
And-
again, it's just a matter of the comparison, right? Because that's what the FDA kind of wanted to see, of just like,
Sure.
okay, hey, is this having an effect versus the placebo group, right? And it's non-selective, right? So it's lowering mutant huntingtin protein and then also normal or regular huntingtin protein as well.
Sure. And I don't know if you saw this because I was reading a good question that Katrina just got from our stream that she'll ask you, but did you say after the year, is it the higher dose or the lower dose?
So then at that point, they'll be randomized into a lower dose or higher dose cohort. So
That's what you said. Okay, I'm sorry. Yeah, okay.
Is there a plan to release biomarker data from the early participants?
So yes, so hopefully there will be an update later on in the year because we have some more data from those participants. At another meeting, you'll probably end up seeing some sort of update from us as far as like where we stand with those participants that have already been enrolled in the trial for a little bit. So yes, so hopefully later this year.
Awesome. Okay. And then the next is, has there been any success seen in the European patients?
Any success seen? Again, we're not really sure, right? We're going to have to go back and look at the data and see exactly what the biomarker question is. A great one, right? Because that's what we can tangibly see. But as of right now, there's no update. So hopefully we'll have that at some point.
Awesome.
Cool. Thank you, Edgar, for coming on with us today.